Welcome to Infection and Immunity Evidence Explained, a Doherty Institute podcast. I'm Rebecca Elliott. The World Health Organization reports that there are about 1 billion cases of seasonal influenza worldwide every year. One type of influenza is Influenza B, a strain that can be overlooked and is understudied despite its significant impact on illness and mortality, especially in children. Researchers at the Doherty Institute have made a remarkable discovery which may revolutionise how we are protected against Influenza B more effectively and for longer periods. Joining me today to discuss this research is Tejas Menon, a graduate researcher at the Doherty Institute and co-first author of this study published in the prestigious journal Nature Communications. Tejas, congratulations on your research and welcome to Infection and Immunity Evidence Explained.
Thanks for having me.
So we recently had Kanta Subbarao on the podcast to talk about the upcoming flu season here in Australia, and we covered off the basics of the virus. So I'll start by asking you, how does Influenza B differ from Influenza A, and why does it remain an understudied virus?
So Influenza A and Influenza B are the main types of flu that circulate yearly and cause our seasonal flu epidemics each year. Flu A is more common seasonally, and it also has what are called animal reservoirs. So they infect a whole host of different animal species like birds and pigs. And that's a real problem because those reservoirs are a potential kind of source for pandemic influenza viruses. They can also cause outbreaks that are dangerous. So, for example, there was an H7N9 outbreak back in 2013, which was quite fatal if people got it. Flu B doesn't have animal reservoirs. It mainly circulates in humans. And because of that, it doesn't really have a pandemic risk. And that's part of the reason why it's so understudied. But it's not something that we should underestimate. It still makes up a significant portion of our flu cases each year. It's on average about 25%. But depending on the season, it can be as high as 60%. And indeed, in Australia, our flu season was about 40% flu B cases last year.
So does that mean that, does it change as much as the Influenza A virus from year to year?
It's a good question. It does change each year, but it's not as quick as Influenza A. And it's part of the reason why it's also kind of underestimated as well.
So Influenza B infections are more severe in children and older adults. I think that's the case. Why is that?
It's not fully understood why flu B is. It's mainly more for children that's really severe. It's particularly school-age children between kind of the age of 5 and 9. The reason behind that's not really well understood. One kind of theory or hypothesis that's kind of been put forward is that we tend to get flu B infections much later in our childhood versus flu A. So it's potentially the case that when these older kids get infected, it's their first time seeing flu B, and so they don't really have an immune response to it that can really protect them from a severe infection. One thing that our group has done in this latest paper that we published is we showed that kids have a lower memory kind of response or memory population for these killer T cells compared to the other age groups, and that might be one kind of indication that these kids are still building up their memory towards Influenza B.
So we know that the best prevention against Influenza B is to get an annual vaccine. Can you remind our listeners why we need to get a flu vaccine every single year?
So the reason why we had to keep getting a new flu vaccine each year is that the flu mutates. And what that means is that each year we have to keep reformulating the vaccine to kind of keep up to date with the strains that are circulating. The reason why we have to do that is because the immune response you have against a strain in a particular year won't necessarily protect you against a strain that emerges in a subsequent year. And so because of that, it's really important that you get the flu vaccine every year so that you have an immune response or some kind of protection against these new strains that might emerge.
So how does this vaccine actually work?
So current vaccines are mainly reliant on the antibody part of our adaptive immune system. So antibodies are these kind of Y-shaped proteins that kind of circulate in our bloodstream. And these antibodies are able to bind onto surface proteins that are found on kind of the surface of an influenza virus. And by binding to these proteins, it can essentially prevent the virus from getting into a cell and infecting it. But it's also these same surface proteins that tend to mutate so rapidly, and they're the reasons why we have to keep updating our strains, because those immune responses kind of become outdated as those proteins mutate.
And why does a universal one-shot vaccine still elude us?
Yeah, it really, again, comes down to mutation. It's really what makes this such a difficult challenge to deal with. The research on universal influenza vaccines are really focused on trying to find parts of the influenza virus that are highly conserved and don't really change each year. And so that's kind of easier said than done, and it's a big focus of the current research on this topic.
And that's the focus of the research that we're going to talk about today. So you recently published a paper in Nature Communications which might change the way that we do get vaccinated against the flu. Tell me about what you discovered.
So in this study, we aim to find new influenza B virus fragments which are targeted by our killer T cells, which are a type of immune cell in the immune system. In total, we found about 9 of these killer T cell targets for influenza B, and that brings up the total number of killer T cell targets to 27 for flu B. And additionally, these targets are also highly conserved in the virus's history, so they don't really change year to year like some of these other kind of targets do.
So how do you go about finding these viral fragments? What's involved in the research that you do?
Lots of screening in the lab. So we have collaborators at Monash University who did a few experiments to kind of make a big list of viral fragments that we could test in the lab. So in total, it was about 112 that we tested and screened in the lab. So that was a big part of my first year of my PhD, was doing the screening methodology. And so after that screening was done, we were able to narrow down onto those 9 conserved fragments that we described in the study.
So as you touched on in your research, you focused on a type of immune cell, the killer T cells. What role do they play in combating influenza B infection, and why are they particularly significant in vaccine development?
So killer T cells are very important in dealing with viral infections. So we in the lab kind of call them the ninjas of the immune response, and the reason why is because they're really good at finding cells that are infected with a virus and killing them before the virus can use that cell to make more copies of itself. And what makes killer T cells so good for vaccine development, or so attractive for vaccine development, is their ability to recognize highly conserved parts of an influenza B virus. And so defining what those conserved parts are is really important for us in the future if we really want to make a more robust vaccine against influenza B.
So how do T-cell-based vaccine strategies differ from traditional antibody-based influenza vaccines, and what advantages do they offer in terms of providing broad immune protection?
So antibody platforms are really about targeting surface proteins of a virus and essentially binding to them to neutralize them and prevent them from infecting a cell. As such, they can prevent people from getting infected. A T-cell-based vaccine can't stop an infection, but what they're really good at is preventing severe disease.
So if we're looking at the influenza B virus and looking for these viral fragments, is that something that we could then look to do for influenza A?
Yeah, and that's something our research group is really working on, and we're making progress in that as well.
Excellent. So what are the next steps for your research?
So now that we have kind of more of these fragments that are well known, we really want to get an idea of kind of how many people they can protect, and so we want to get an idea of what kind of global coverage we can get with these fragments.
You said that there's now 27 viral fragments that you've identified. How many could there possibly be? Do we know that?
It's hard to say. I think there's definitely more work that could be done in that sphere. But in terms of an upper number, it's hard to know.
And I always have to ask this question, is that how far do you think we would be away from a one-shot universal flu vaccine?
That's a very common question that I've gotten since the paper was published. The answer is really hard to know. We can't really put a timeline on this kind of thing. But the work that we've done here is definitely a step in the right direction.
While we wait for a universal one-shot vaccine, we've got the annual vaccine. Is there any message you want to tell our listeners?
Yeah, it's still important to get vaccinated for the flu yearly. We don't really know how far away universal vaccine is. So for now, our best kind of protection really is the vaccine that we have right now. So that's really the one that people really should be relying on.
Excellent. Well, thank you so much for joining me today on Infection and Immunity Evidence Explained, Tejas.
Thanks for having me.
And thanks to all of you for listening to Infection and Immunity Evidence Explained, a Doherty Institute podcast. If there is a specific topic you would like us to cover, please get in touch with us by emailing doherty-media@unimelb.edu.au. That's doherty-media@unimelb.edu.au. If you've enjoyed our podcast, please rate and review on your favourite podcasting app and subscribe if you haven't already. The Doherty Institute is a joint venture between the University of Melbourne and the Royal Melbourne Hospital. See you next time.